Welcome back to our course, Machine Learning for High Throughput Biomedical Data. In this lecture, we will discuss some basics about exploratory data analysis that includes some visualization and some summary of data, as well as simple transformation and normalization techniques for gene expression data. We decided to add this lecture this week as it is important to prepare the data for a theme that we will explore throughout the week, classification. During this lecture, we will rely on box plots, scatter plots, and bar charts to explore our data. As a reminder, let's take a look at the box plot and what kind of information it shows relevant to our analysis. The box plot uses a simple separation of all values into quartiles, or quarters. It shows the top 25% of the values, or the upper quartile, lower 25% of the values, or the lower quartile, minimum and maximum values, as well as the median and outliers. The box in the middle represents 50% of the values, quartile 2 and 3. Okay, so now let's open our data set and take a look at what we have inside. As you remember, we originally prepared 11 samples. Approximately half are normal-like and half cloud and low. You can see the gene names on the left and sample names on top. I highlighted the ones that belong to normal-like samples in green and cloud and low samples in yellow. To compare transformation techniques, let's already prepare a table of the same expression values but converting them into logarithmic scale. As you remember, the rna-seq pipeline that we ran produced this expression genes in FPKM values. We are going to take this file and let's open it up in Excel. Okay, so we have the file in Excel, and now let's take a look. We have the gene names on the left and the sample names on the top. Now, from what I remember, these samples were normal-like, and these samples are cloud and low. Let's just mark this in bold so that we can remember. Now we started formatting, so let's go ahead and save this as Excel. Now, as we're talking about transformation, we're going to be preparing the same exact table, but in logarithmic scale. How do you do that in Excel? Well, first of all, let's copy the header, place it over here, and now there's a function in Excel called ln for normal logarithm. Remember that you can't take a logarithm of 0. So what we will do is we will do equals ln, and here we will take a number and add 1 to it. And now we will copy this, and we want to do paste special and copy paste formulas. Okay. So now we have the logarithmic scale of this column. Now what we can do is we can select all of this. Okay, so now we have this column. This column is all of the numbers from the previous table, but in logarithmic scale. And we are going to start looking at what would be the difference between these values and these values over here. Now, just so that we can keep everything together, let's write for ourselves that this is now ln scale. Okay, the other thing that we want to remember is that we measure things in terms of difference, in terms of what kinds of differences do we have here. Well, we have the p-value. If we use t-test, it will be p-value, our statistical significance, and we use fold change. Let's quickly add that. So this will be t-test, and this will be fold change. In fact, it's going to be log fold change. And fold change oftentimes is represented by FC. So let's just add log FC. Now if we do t-test, we can do just t.test, and we can take the first half and the second half. Then you can write 2, 3. And we can, again, copy it all the way down. Now, what is fold change? Fold change means that we take an average of our first group, and then we divide it by the average of this second group. Okay, so that's going to be our fold change. Average of this divided by the average of this. Now we get a pretty big number. Let's now paste it all the way down. Okay, but we said that we wanted to be in logarithmic scale. So instead, let's add here logarithm scale of this. Okay. Okay, so now we have log scale of fold change and t-test. So how do I know what is highly differentiated and not so highly differentiated? I am always going to be looking for a p-value that's going to be approximately less than 0.05, so less than 5%. And log fold change, oftentimes we look for the ones that are more than 2. So for example here, if you just take a look at this bar chart, let's quickly insert here a bar chart. Okay, and we can see that the first 5, so 1, 2, 3, 4, 5, are going to be much higher than the rest. A good way to explore data is to perform summary statistics. If you are using Excel, you can use the Incorporated Data Analysis add-on and run Descriptive Statistics option. As a result, you will get several ideas about the values of gene expression for each sample. In the resulting table, you can see such things as median, minimum and maximum values, and skewness of data. You can see that the median is right about 23 to 22, and skewness varies by sample between 12 and 20. We can visualize these values in a bar chart to see how they compare between samples. When we visualize these values and see that the median and skewness vary between samples and raw data. Now let's take a look at how to create a box plot. So what I want is to take all of these and I want to insert a box plot. So I go to Statistical and I go to Box and Whisker. Now it takes a little bit of time, and now I have my box plot. Now I can modify the look and feel, and I can show you how to do that later. But let's at the same time, let's first of all move this up to the top. Okay, and let's insert it here. We'll not worry so much about modifying the look and feel just yet. But now we can go over here, select all of this, insert, and again, Statistical, Box and Whisker. So let's now compare these two charts. So let's take it up over here. Okay. Okay, do we see any difference between these two? Well, we do. They look completely different. We can see that the upper quartile dominates this side, and here it is normalized. Here we have the middle part, the median is aligned pretty well, and this part is, of course, is not so dominant. So what is that? That is called skewness. So what measures do we have for identifying this characteristic of the data set? The way we can do this is by using a tool that is inside Excel to give us some summary statistics. So we can take these box plots and move them over here. Okay, and now let's do some summary statistics. Go under Tools and go to Data Analysis. And here what we can do is find descriptive statistics. Click on OK. Let's take as the range the first non-log transformed table. Okay, we'll select this data set. Labels in first row. Summary statistics. Let's click on OK. We will wait a little bit. And now we have a new sheet that shows us all of these values for each one of the samples. Now, for us to make it easier to work with this, let's rearrange this table a little bit. So if you can see, each one of these actually repeats, so I don't need to keep them, so I can delete them. Okay, and now it's easy to compare. So how can I see the mean, for example? I can insert a bar chart, and this is my mean. I can do the same thing for median. Let's do the same for the second set of values that are log transformed. So now if I go over here and I go again to Tools, Data Analysis, Descriptive Statistics, okay, let's select now this as my input. So now we can add another series. So this is going to be mean raw, and we'll add mean ln. And now we have a mean. Okay. So our mean is way low. Let's take a look at skewness. So this was skewness raw. And we'll add a skewness log scale. Okay. Okay, interesting. So the skewness is different. And of course I can do the same with the other values that I see here. Now this is a very small scale, so why don't we do this separately and just take a look at what we see in there. Okay, still exists, but it's a range between 0 and 1. Okay, so this is some summary statistics. Now we are ready to do the box plot, take a look at it, and how to modify it and make it look exactly the way that we want it to look. Okay, so basically what we want is we want to remove a few things, we want to move things around a little bit, and then we'll be ready to compare these and will look much better. So let's go into Format, and let's open the Format pane. And now you have the gap width. You can just set it to to 0.1. That will just spread them out a little bit. let's do the same thing over here, 0.1. This is now a much better looking box plot. Let's change this to ln scale cell line expression. Gene expression levels are heavily skewed in linear scale. Majority of the data points, the lower expressed genes, have very low values, and the rest, the higher expressed ones, between 1 and very big numbers. In our case, you can see that the majority of dots we observe on the box plot range all the way up to 16,000. If we transform our values into log scale, we see a much more normal bar plot. Now I can compare values between samples without losing the detail of lower expressed values and still see the top 25% of the values clearly. Another useful transformation you might want to consider for NGS data is quantile normalization. In order to compare your samples, you might want to align their medians in the following way. Here under Utilities, you can see a number of different tools that are useful for these kinds of activities. You can go under Utilities, upload the same file. Let's upload expressions genes FPKM, and then we will click on Define Groups Check. We don't need to add any groups. Let's click on Start, and then select Quantile Normalization. So what are we expecting? We're expecting the median, that line in the middle in the box plot, to be aligned. Okay, so we do want to transform the table to logarithmic scale, and we will use a threshold of 6, which is kind of a cutoff for a lot of next generation sequencing expression values. Here, we'll just name this as Quantile Normalization for cell line expression data. I'll save it, and I'll run the pipeline. Let's look under My Pipelines. And here I actually just prepared the same exact pipeline right here. I can go to Download Output Files. And now I can see that threshold 6 normalized. So I want to see this file, and I can go to my downloads. Let's go to Downloads. And here is my normalized table. Let's take a look. What does it look like? Okay, well, it's giving me some numbers. Let's try and select all of this and create a box plot to see what it looks like. Okay, well, what do we see? As expected, we are seeing, let's format this real quickly just like we formatted the other ones. Alright, this is what it looks like, exactly like we wanted. This makes for a better comparison between samples. It is important to note that the transformations we performed on the data actually do not change the overall structure of the data. For example, if we do PCA analysis, we will still see a clear separation between groups of samples. However, differentially expressed genes will change. For example, I can count the number of differentially expressed genes using t-test and fold change, and I will see that the number of selected genes using the same threshold will be very different. What is important is to keep in mind what parameters to use to determine the significance of the differences between groups of samples. Now let's talk a little bit about interpretation. how do we annotate gene names, and discover their function. On TBioInfo, there is an option under Utilities to run an annotation pipeline. This annotation pipeline will convert your E and S G values into common gene names and provide you with gene ontology description for each name. Today, we learned about data exploration and transformation that is useful to prepare it for analysis. Now that you've learned how to do this and how to explore your data, play around with the full dataset that contains all of the different subtypes. To learn more about the subtypes and the importance of classification, please read this publication.